All right, all right. Welcome back. I am Kevin Curran. This is the second in a video series on delivery of gene editing tools. So, this is one of the big challenges of the next decade. It's 2024 right now as I record this video. We have gene editing tools commercialized. We have an FDA approved CRISPR-Cas9 drug for sickle cell disease. The big challenge now is to learn how to deploy these gene editing tools all throughout the body, right? Vertex has demonstrated they can effectively deploy CRISPR-Cas9 ex vivo once cells are removed from the bloodstream. Bone marrow stem cells can be gene edited in a lab with good efficiency and then infused back into the body. And the developed red blood cells produce sufficient hemoglobin to make this a good therapy for sickle cell patients. That's great. How do we move to in vivo administration, which is a simpler route where we just inject into the bloodstream systemically or localized into some part of the body, the eye, the brain, the ear, or reach the cell type through the bloodstream, right? Different strategies, every disease will be different. In the earlier video, I reviewed the use of viral vectors and naked cargo, right? So using AAV viruses or lentiviral vectors, so sort of taking advantage of the natural transduction capacity of viruses to bind specifically to certain cells and then deliver into the cell, deliver the cargo. And then the use of just deploying Cas9 protein bound to guide RNA, what we call RNP, ribonucleic protein. I'm calling that naked cargo because we're not wrapping it in any kind of a vesicle. We're just injecting it into the body. And that has a lot of challenges, right? So that has a lot of challenges in terms of editing efficiency. The viral vectors have challenges because they could lead to long-term expression of Cas9 nuclease. This enzyme could be, active, chopping up DNA for longer than we want, and that could lead to safety issues. So there's been a lot of interest in lipid nanoparticles. That's what I'm going to talk about today on this video. And maybe let's see if I can keep it to 10 minutes. Let's see. It's stranger things have happened. I can be concise. I'm going to talk about LNPs, and then in the final video, it will be a very short video on VLPs, virus-like particles. All right, folks, let's keep it moving. LNPs, lipid nanoparticles. These are synthetic balls of fat, proprietary synthetic balls of fat. Lots of companies have been developing their own LNPs. LNPs, the name's very descriptive. It's a ball of fat, and they're on the nano size in terms of diameter. They will deliver RNA and RNP. So, we can think about this historically. In 2018, Alnylam, a Boston company, they got the first FDA approval for a drug that relied on LNPs for delivery. In this case, they were using little pieces of RNA we call siRNA, short interfering RNA, and they were deploying the RNA into liver cells to disrupt a gene. And that drug is called Onpattro, and that was a big success for LNPs. As I'm going to talk about today, LNPs are very adept at reaching liver cells and transducing. They're very adept at delivering gene editing cargo into liver cells. And it is a challenge to get them to move off of that default mode and start docking up and deploying cargo into non-liver cells. So that's a big focus of the industry right now. LNPs became worldwide famous during the pandemic because the Pfizer and Moderna vaccine both employed LNPs. Another form of RNA, this was messenger RNA, was packaged into LNPs, and that was the RNA for the spike protein. And in that case, it was a localized delivery, right? The LNPs, they moved into our sort of arm, right? The muscle tissue and the surrounding cells, and that sort of localized deployment into antigen presenting cells and muscle cells was sufficient to cause immune memory. So 2 success stories with LNPs, and there's many companies now pursuing clinical trials with their own LNPs. It is, I like this image. The LNPs, you can think of them as composed of 4 different lipid molecules. There's this PEG lipid in green, the cationic lipid in red. This is a very important kind of critical lipid. I'll mention in a second. There's also helper lipids and cholesterols that help kind of package this membrane together. And then the RNA or guide RNA formulated with Cas9 protein, the RNP, can be packaged inside the lipid. And this cationic lipid is important because once this LNP docks up on a cell, like in a liver cell, it's going to endocytose. It'll enter into the cell, and then the pH inside the cell will engage with this cationic lipid, and that will release the membrane. So the membrane will open up inside the cell and release the gene editing cargo. In this case, it would be guide RNA and Cas9 messenger RNA that can then form a complex and move into the nucleus. Okay. All right. So as I mentioned, clinical work targeting liver cells has been successful. There's 2 reasons for this. One is just sort of kind of a function of the mechanics of our body. If you inject a LNP drug into the bloodstream, it's going to naturally accumulate in the liver because the liver is there to help purify blood. So blood passes by those liver cells. You'll get a lot of gene editing LNP drug on the surface of the liver cells. And then why would the LNP dock up and enter into the liver cells? Well, lipids will naturally bind to this ApoE protein that's in our body. ApoE binds to lipids, so it's going to bind to the surface of lipid nanoparticles. And now you've got an LNP bound to ApoE. ApoE can specifically bind to LDL receptors, which are heavily present on liver cells. Okay, so here's an LNP, and you can see there's ApoE protein on the surface. And now we're looking at a liver cell. This ApoE is going to bind up with the LDL receptor and then be taken in. See this, the membrane will endocytose, and now you've got the LNP inside the cytoplasm of the cell. Now there's a pH change inside of this vesicle, and that engages with that cationic lipid, and the cargo is released. So, like I said, now you've got the gene editing tools inside the cell, right? Think of this like, armies moving in through the Trojan horse. And you get this liver cell specificity through the docking of ApoE against the LDL receptor, okay? So this is working well. And so when you look at the landscape of pharma companies pursuing gene editing drugs, a lot of them are in the liver, especially a lot of the late stage trials, because liver cell is attainable. So we can look at Intellia. Intellia has as far as I know, the most advanced clinical trial, most advanced gene editing trial with in vivo delivery. Okay, so they're now dosing for a global phase 3 study for NTLA 2001. And this is for a type of amyloidosis called ATTR. The idea here is you've got a mutated TTR gene, and you want your CRISPR-Cas9 to bind to that gene and disrupt it by inserting indels, right? Big insertions and deletions to knock out that toxic TTR before it forms these plaques that would be really detrimental to liver cells. So you can see phase 2 data over here. 60 patients showed deep, consistent reduction of the toxic TTR after a single dose of NTLA 2001. So that's great. That's why the FDA greenlit this to advance to the late pivotal study, right? And so, this is really just a clinical example of the LNP description I just gave. Intellia is packaging single guide RNA with Cas9 mRNA into a lipid nanoparticle with ApoE proteins. So there's their LNP, and it's going to access the liver through this hepatic artery. Something to note here, they're using 2 RNAs, right? Guide RNA and Cas9 mRNA. The approved drug for sickle cell disease by Vertex, Casgevy, they pre-formulated Cas9 protein and let it bind to guide RNA. And then they electroporated that into bone marrow stem cells outside the body. So again, I want to emphasize that depending on the disease, depending on the route of delivery, you can go with 2 sources of RNA, like Intellia is doing, or you can form the complex, the Cas9 guide RNA complex, and then move that in. Okay, so different companies that are going after different diseases are using different strategies here. So this is Intellia. I like this image. Gives you the whole picture, right? So here's Intellia 2001 hitting the surface of liver cells. It's got ApoE proteins binding to LDL receptors. It's endocytosed into this endosome, and then that's where the LNP breaks down. Now you've got the Cas9 mRNA will translate in the cytoplasm into the protein, which will then form the complex with guide RNA and enter into the nucleus. Okay, so here we're sort of co-opting a lot of cellular machinery that naturally exists in these liver cells. And then once we're in the nucleus, this is sort of classic CRISPR-Cas9 activity. Here's the Cas9 enzyme bound up with single guide RNA. That complex will first identify this short PAM sequence and then complementary bind through the guide RNA. So that's 2 points of kind of identity with genomic DNA. And then the Cas9 delivers the double strand break. So you're breaking apart the chromosomes, and then you let this natural editing technology to occur, which is called non-homologous end joining. This is the cell trying to fix a double-stranded break, and in the process, it's introducing in mutations, indels that disrupt the TTR gene. So all of that is happening at this targeted TTR gene in liver cells. That's how Intellia is disrupting TTR. If they can knock this toxic mutated TTR out of these patients, they should see reductions in the disease. Okay, so this is working in liver cells. And so you can imagine a lot of companies are going hard at genetic heritable diseases in liver cells. The challenge then becomes, how do we move outside of liver cells? Imagine if we could directly inject gene editing tools in some kind of a LNP and have them reach the bone marrow stem cells, right? We could go after all of these heme diseases, blood disorders, without the ex vivo process, that cumbersome process of performing ex vivo therapy, right? So there's a lot of groups trying to tweak with the surface molecules on LNPs to go after circulating peripheral bone marrow stem cells. So that's kind of the systemic delivery piece. You can also think about LNPs. Maybe we can avoid entering and docking up on liver cells through localized injection. So this is a common strategy. You're gonna, that's how COVID vaccines work. A lot of this is happening. A lot of this is still in early phases, a lot of preclinical, but in mouse work, folks are going into the retina, the cornea, and the eye, the inner ear, the lungs, the spleen. But you can tell, as you follow these companies, we're still figuring out optimal formulation of LNPs, how to tweak with these synthetic lipids to gain cell access, but to not trigger toxicity. And I mentioned another company called Verve. They've been using beam editing technology, and they ran into a spike in liver enzymes with one of their LNP systems, and so they're trying to kind of shift focus and try another LNP. I think that just drives home this point. We don't have LNPs totally figured out, and I think the main issue here is that this is totally new. This is synthetic. These are not naturally occurring lipids, and so the body is going to have some kind of reaction to this. The liver is going to struggle to process this or immune rejection. I think all of these pieces are sort of still being sorted. Speaking of sorted, I wanted to highlight one paper that shows kind of where the progress is being made in terms of new LNPs. This is in mice, but this group here published on this sort technique in Nature Nanotech. They are injecting LNPs into the tail of mice, and then they're tracking to see where they end up. So they're imaging here. You can see delivery to lung, here to spleen, here to liver. And they're putting in a new, customized sort lipid here that will give access to these LNPs. So here's one example. They added a cationic lipid called DOTAP, and that was sufficient to target the organ, to target to lungs instead of liver. Okay, so progress is being made, but we're still many years out from being able to deploy LNPs into non-liver cells through an in vivo delivery. We still need to figure out new LNP formulations, take into consideration safety and tropism, cell-specific binding. And this will probably be just a lot of high-throughput screening of unique formulations. It may be that we don't fully understand the science of a lot of these lipids and formulations, but we test them enough, in cell culture to see how they behave in terms of cell tropism, and then figure out, the safety piece with animal work. I think that's plenty for now. I'm going to wrap that up. That was kind of a crash course in where we're at with LNP delivery of gene editing. And the final video in this series, which will be brief, I will touch on virus-like particles. All right, folks. Subscribe, like, share. Like I said, if people are enjoying these videos, I'll keep posting them. I like to talk. I like to teach. So let me know. Okay. Take care.